Okay. John, I've been told I have to stand next to you to introduce you. There you go. Great. So, hi everyone and everyone at home. It's my great pleasure to be standing here introducing John Novembre as our seminar speaker. John is a professor at University of Chicago in the Department of Human Genetics and Evolution and Ecology. Among other things, John, among many laurels and career highlighted by really insightful work in spatial population genetics, I'd like to embarrass John and point out that John also in 2015 was named a MacArthur Genius Awardee. So we have a real genius in front of us. And I like to embarrass him that way because I've known John for a really long time. John and I met when we were in our first year of grad school, 22 years ago, approximately. And when you see one of your peers succeed in this incredible way, in this sort of like stellar way, it's just amazing. It's just amazing. So, something that, you can kind of express pride for a little bit. So I've always been incredibly proud of you for having done that, having gotten that. Yeah, I'm not going to blather on. John, really excited for you to be here.
Thanks so much. Thanks so much. Yeah, it's funny, as Andy mentions that, I don't know if I've ever told a story, but from that time where we were both in California and I was a graduate student, I remember meeting Russ Landy and finding out that he had gotten a MacArthur Award. And I thought to myself, oh, that's just amazing that people in this field can get that kind of recognition. And it just made me appreciate like what I was doing, sort of like this evolutionary biology stuff. Some people care. It's not so esoteric. There's a group of people who are willing to reward it, I suppose. And anyway, so I hope some way that might make you feel like this is an important area to be working in.
Hold on a sec. There's something weird with the audio.
And John, you might take off your mask.
Yeah.
Might be easier.
Mm-hmm. Oh.
Hello, hello. Thumbs up if you can hear us.
Or say something. Hello. Back to the.
It was better before.
Hold on. Say some stuff, John.
Hello, hello. Testing, testing. Testing, testing. Testing, testing. Testing, 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 testing. It's not very loud, is it? This one.
Well, it's not, that one is not supposed to be loud.
Testing, testing.
There you go. That's good.
Okay.
Better.
How about that?
No.
Hello, hello, hello. Hello, hello, hello.
No, it's been amazingly good.
Hello, hello.
Say some.
Hello, hello, hello. Hello. Test, 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 test. Test, test, test. Go the other way. Input volume low. Maybe it'll. Test, test, test. Test, test, test. Hello, hello, hello. Hello, hello, hello.
Hey, Zoom people, can you hear us?
Yeah, it's pretty weak though.
What the hell?
It's this one. Is the yellow, is that normal color?
Yeah, that's normal.
Okay.
This generally works on people's. Let's see. How about Zoom? Is this? No, it's being very.
Sounds far away. Okay.
Well, let's go back to this.
Hello, hello, hello. Is that better?
Well, we're not using this one, so.
Yeah. Yes, it is. Okay. I'll stay close to the laptop.
Yeah, and it's going to be a bit echoey, but then we can do about it.
It'll be a bit what?
Echoey.
Oh, I see. Okay. All right, so it's better and we'll continue. Great. Okay, so, oh, is that the echoey that you mean or? No,
No,
that's just
you won't
loud.
hear it. It's
Yeah.
over Zoom.
I see. Okay, great. Okay. Okay. Excellent. So let me get where I can see my slides. Okay, great. So today I wanted to give a talk where I give a background to some of my lab's research interests or from the broad perspective, and then zoom in a bit and talk about 3 problems that we've been working on recently. And to introduce my lab, this is a sort of old lab trip that we took to go ice fishing in Milwaukee. This is Hussein Al-Assadi on the left, who wrote the map software that you may know. A very avid fisherman, and year-round is excited about fishing, and he took us fishing. That's Joe Marcus, who looks like he's out of the remnant there on the left, and we go to Mark Rappel. And so this was maybe circa 2015. We've leveled up lately, got a sailboat now, and life is a little bit better. And so yeah, we live along Lake Michigan in Chicago. And in terms of introducing us, I want to kind of contrast the tools that we use, which are from computational statistics, theoretical population genetics. So these are the tools, the fishing rods, depth finders. And the questions we ask are from the areas of evolutionary genetics, and also human genetics and genomics. And the questions we're asking are kind of more biologically motivated, push us to explore new areas of computational statistics and theoretical population genetics that we might not have understood before. And then sometimes it gives us insights on questions we can ask that we never thought about asking, what is this feedback? And I imagine very similar things happening in Andy and Peter's labs too with this sort of skill sets. The goal is usually like analysis tools that will empower other researchers, but we also like to work in specific empirical studies to help build our expertise. So one of those areas for me that is a now long-running collaboration with geneticists in Sardinia, and where there's a mix of complex trait genetic studies going on, but we're bringing in a population genetic perspective. So we've been looking at things like evidence of polygenic selection on human traits like stature, multiple sclerosis risk, and then building demographic models of Sardinia so that we can calibrate our measures of selection. And that has led us to developing an ancient DNA project in Sardinia that we published the first round of that. So this is the collaborators from that work, and I actually got to go out in the field. There's an archaeological site underway, and that was involved in bringing samples into the study. So it's been really fun to be involved in that. Then another area is working with non-model species, wildlife species, and canids in particular, from a long-standing collaboration with Bob Wayne at UCLA. So we were involved in some of the first whole genome sequences of wolf genomes, doing inferences of natural selection and the evolution of domestic dogs, and relatively recently, mutation rate estimation from pedigrees of wolves from Yellowstone National Park. When we're looking at sort of methodology development, there have been sort of 2 major forms of structure that we focus on in genetic data. One of those is haplotype structure. And so some of the cool stuff we're up to recently is work on inferring runs of homozygosity in extremely low coverage data. So even from what is called pseudo-haploid data in ancient DNA, where you actually just have like 1 read for any location in the genome. And you might think you can't infer runs of homozygosity if you only have an observation of 1 allele, but actually the LD patterns can allow you to see it. And so that's work with my former postdoctoral Ringbauer. Then with Arjun Navanda and Matthias Steinerking, we've been looking at 2 locus genealogies in LD and temporally structured samples. So this is also ancient DNA inspired, and trying to understand basics of LD and haplotypes in ancient DNA, and some cool stuff there about like if you know haplotype copying models, how the jump rate changes as you look at older and older samples, and that the slope of like how much the rate increases with time, actually, or how it changes through time, it shifts dramatically depending on the demographic history of the population, which is kind of interesting. And then with my former graduate student Diego Ortega-Vecchio, I'm working on using haplotype lengths to infer selection coefficients for both advantageous and deleterious alleles. So usually people think about like using haplotype length statistics like IHS to detect sweeps, but there's also a signature around deleterious alleles. that can be used to infer selection. So that's haplotype-based projects. It's kind of a cluster of work and theme that we've had running for a long time. The other is spatial projects. And so these are things like the migration map projects that we've worked on, these migration surfaces. And we've got a recent paper out on sort of fast inference of these surfaces, where like the blue areas represent areas of high effective migration and dark areas of low effective migration, and they help visualize patterns of population structure across the landscape. And then this project I'll actually be talking about a bit in this talk, so I'll slide right over that. But yeah, so today we're going to dwell in this topic area of spatial genetics, and I wanted to tip the hat to a lot of the work that's been done here, and in terms of those who are not yet exposed to it. There are some great reviews from Gideon and Peter, from CJ, Peter, and Andy, and then I threw in one from the last one that I was involved in writing, now quite old, but it's, I think, a very exciting field and exciting to see like more momentum building in this area. And so I want to tell you about sort of these three problem areas that we've been looking at and some of the results that we have. So one of the challenges that I've noticed in working in this field is that there's a lot of development of algorithms like this is results from the structure algorithm, where it's stacked bar charts that represent individuals' levels of membership in source populations in terms of their ancestry. There's PCA type plots, tree mix type plots, those migration surfaces, and many other UMAP type plots, right? That all are meant to represent this idea of population structure or some kind of genetic structure in the genetic ancestry of the individuals. And we do that to try to understand, the basic basic properties of our data set. It's oftentimes one of the first exploratory things that we do. But a major pitfall of all of them is that they don't convey on an absolute level of sense how different the populations are. So these populations can be very similar to each other in an absolute sense, of the allele frequencies being just slightly different in any of these source populations or across these gradients that PCA map or across the most diverged populations of this tree, and you don't get a sense of just how diverged they really are. There are relevant statistics you can compute like FST between the source populations, look at the proportion that it's displaying, look at the drift parameter, but none of these really leave you with a sense of like, what are allele frequencies actually like out there in nature? And so that's motivated us to think in this area and work a little bit in it. One of the things you can do is talk about the proportion of variance explained. So, and that has a long history going back to Lewinton and these really impactful, important paper on the apportionment of human diversity, where this very famous and important statistic of that less than 15% of allele frequency variation is found between populations, most of the variations within populations. That's really important for people having some sense that there's not very large levels of absolute differences between human populations. I'm flagging here, I was part of this special issue of the Philosophical Transactions of the Royal Society that was celebrating the 50th anniversary of Lewinton's paper. So I wrote a kind of history of science type piece in this, and it's a nice, I think, I enjoyed writing it, I won't say more than that. But yeah, this is kind of hard to get your head around, right? Like, and really understand. Now, one way you can try to teach it is to relate it to Hardy-Weinberg ideas and plot genotype frequencies as a function of allele frequencies. And the p squared, right? P squared is a parabola. There's a y equals x squared. There it is. 2p, 1 minus b, and the 1 minus b squared, those are those 2 lines there. And then you can see how in certain scales, the heterozygosity, let's say focused on the green line, is quite close to what you would expect under band, right? And in other scales, you might see some divergence. And in humans, the heterozygosity, at the global scale, the heterozygosity is reduced, and it's reduced about 10%, and that's about what you see for FST. So this FST, which is also related to proportion of variance explained, is somehow related to how far we are from random mating expectations. And in this kind of view, humans are not that much, different from what we expect in party one group, the random remaining population is 10% reduction. But still, you don't really get a sense of like, what do allele frequencies look like across populations, right? Another way of showing human differentiation on an absolute context.
There's a vote for you to stay near-ish to the computer.
Yes, okay, great. And
It's doing fine and don't rate, and we're fine with the time. So
okay. Okay, great, thanks. And then another way is to kind of like look at human differentiation from the scale of our sister species and the levels of differentiation. So this is a figure that came out when I was in graduate school and found it absolutely stunning and still love it today. It's from some of the earliest sequencing that was done of multiple individuals within species and between species in primates. It's a section of the X chromosome. And you can see levels of differentiation between gorilla samples, orangutan, chimpanzees, bonobos, and then between 72 human samples. And you see, wow, just how remarkably similar the genetic diversity is amongst humans relative to our primate neighbors. So you wouldn't think that two gorillas or two chimpanzees would be maybe more differentiated than we are, given how much phenotypic diversity we seem to show on the surface. But there's clearly something more complicated going on, that we have such average low levels of divergence, and yet a few phenotypes that seem really different, right? So that's helpful. And this is just to say that these results have kind of held up in the age of whole genome sequencing of primates. So that's results from the 2013 sequencing paper. So, but again, still, are allele frequencies in humans like sometimes 0% one population, 100% another? Or are they all kind of bouncing around the same levels? Like, what does the allele frequency variation actually look like? How can we, convey that and teach that better? So that's been the goal of this kind of set of projects. And oops, I got my animations backwards there. But anyway, it's these two projects that are kind of dwell on this theme. And the first one developed this browser of allele frequency variation, just a simple resource tool. I think this is a place that appreciates the work, the value of developing tools for the community. And so this simply allows you to look at several major human genetic variation data sets, put in a SNP, get a map, hit save as a PDF, and maybe put it into a figure in a paper or a presentation. And there's some tricks you need maybe to play, because in large human samples, many variants are very rare. And they would be tiny little pie slices, right? And very difficult to read. So we have this kind of notion of like a scientific notation for like order of magnitude for pie charts, where like sometimes we scale the pies so that they are out of a much smaller denominator. So here, this is 25% of 1% is 0.0025. So you can see this is 0.5% right here and absent everywhere else. Okay. And so the legend changes, and there's a color scheme that's associated with that. And there are a couple other fun tricks, like we use transparency to indicate like varying levels of uncertainty because of sample size differences. Okay, so we put in a random button onto this browser, so you could just click random and see like what some random variants look like. And that just seemed like something fun at first, but then we realized that this is actually a very teachable, like useful teaching tool, because one way to gain some understanding of a system is to look at, an ensemble of random draws. And so this is a little animation of hitting that button, I think about 10 or 15 times. And so this first one is 0.5% frequency in a population in Southeast Asia. And then I'm going to let it, I might need to, there it goes. Okay, yeah. So it's about 1% in East Africa, that variant, about 0.5% in Southern Europe, about 0.5% up in Southern Asia, and then something that's pretty common across the whole globe. Then something common in Africa, then like seen a little bit in populations in the Americas, also like a handful of populations in Africa and the Americas, 0.5%, handful, 0.5%, sprinkled around, you're getting the picture. Okay. These very rare variants that kind of pop up, and something that's sort of handful of African populations in the Americas, 0.5% Southeast Asia, a couple of populations there. Okay, that's it. All right. One thing about those ones that we're showing some presence in some of those, especially West African populations and the Americas. The Americas, this is the 1,000 genome sample, so it includes populations of African Americans in the U.S., and from Central America. So some of that's from the African ancestry that's shared between those places, right? So that's a snapshot of some variants, right? Gives us a sense, wow, there are a lot of rare variants in the human genome. It was kind of interesting. The one common variant we saw was found everywhere. How often, was that abnormal or is that pretty common? I don't know, right? And so, so we want to explore that a little bit more. The general lesson about like rare versus common was something that emerged in is kind of appreciated generally, but I just wanted to this happens, it's an old favorite figure, where I was involved in a project that got to sequence 202 drug target genes in 14,000 people. So it was one of the first deep sequencing studies in humans back in 2012. And this is the raw numbers of common variants per gene versus the raw number of rare variants per gene. And so the kind of tease was that like common variants, the kind of stuff on genotype arrays, is like the tip of the iceberg of human genetic variation. But this plot has nothing about geographic variation. Again, like how do we see like how what are typical geographic patterns and allele frequency variation? So we want some way to do a summary genome-wide of the geographic patterns. And so we might like bin things into these discrete categories, like say, okay, that thing is like, it's rare in Europe, or sometimes it's like rare globally, but like it's rare in that, that population in Europe. This we say, oh yeah, that's something that's common globally. So we want some kind of shorthand that would mirror like what, the investigator might look at a sample and understand. And so, and we also kind of have a notion in mind to like tease apart like these continental, categories that are so often used. Like, do they actually, have any emergent existence in the data? And so what we're doing is take an allele frequency map and put a 5-letter code to represent for each region, whether the variant is common, rare, or unobserved. The threshold for common being 5%, which is so often used in human genetics. And we've played with different thresholds. So this variant, this one would have CCCC as its, like the summary of the pattern, okay? And this one would be UU for unobserved, R for being rare in South Asia, and then UU. Okay, so it's coarse, but it's, there's something meaningful to say that something's common versus rare or absent, undetected here. Okay, so now we're going to take all the variants in the 1,000 genomes data, give them a 5-letter code based on their pattern, and then count how many of each pattern would you see. And think for a moment for yourself, what would you think to see? Like, what are the common patterns that you might see in human variation, or any data set? Think about your own species. Okay, I'm going to show you the answer here. So this is sort of the answer. We've done this as a stacked bar chart. Okay, so on the left here, just to give context, the 2.9 measurable sites in the human genome that were surveyed in the 1,000 genomes data set, 3.1% of those are variable. Of those variable sites, this is how many fall into each of these different categories, and they're rank ordered. So the most common pattern is rare in Africa, absent everywhere else, or undetected everywhere else, and 16% of the variants fall into that category. Then you have rare in an East Asian population, rare in a South Asian population, rare in the Americas, so on, and rare in Africa and the Americas. Then you get up here and you get to variants that show that pattern of there being common, not just in one population, but in all of them, okay? Then a few where it's rare in kind of pairs of populations that have had some connection, and then rare or common in Africa, absent outside, okay? And all this is like an indicator of our human demographic history, but in a more variant-centric way, right? And remarkably, of the 243 possible categories that exist, 7 of them explain 85% of all these variants, okay? And so that maybe is a unique picture of our human genetic structure that you don't get out of a PCA plot or a structure plot. And you might ask why, why is it that pattern, not something else, right? Where are the things that are like common in one population and absent everywhere else, right? Those seem to be extremely rare. Is there like other histories in which that would have happened, right? Maybe, but yeah, the history that we have is that we were a population in Africa that spread out across the globe, right? And so that has, it's a bit green here, but the consequence that, to kind of give it briefly, like any variant that's common in, or most variants that are common in any one part of the world, to be common, it takes time to reach that frequency. And any variant that's that old predates the split. And so if it would predated the split, then it got taken everywhere in general, unless it got lost in the bottlenecks. So we have lots of variants that like pre-existed in Africa, got spread out across the globe that were common. Then we have some variants that got lost. And then we have lots of variants that pop up afresh because they're new mutations from these people who spread out across the globe. But crucially, they're new, they're young, they're clustered geographically, but they're not like common in that area because they're the kinds of things that are carried by, our great grandparents or something, and they're just located where the the descendants of that great grandparent are, right? So this is another way of saying it, like this is simulations with 2,000 generations of divergence, things that were sort of common 2,000 generations ago with sort of human type population sizes will tend to be common in a bunch of independent daughter populations. And then things that were maybe ancestrally rare, they're often lost. And then things that are, new mutations will not reach very high frequencies. So there aren't a lot of things that are common and private to one region because there just hasn't been enough time. We're too recently diverged. This, you can kind of play with theoretical models. So this makes that point even better. Theoretical models where you stretch the divergence time and make it quite deep relative to the coalescent time, you start to see these common private variants. But in humans, we have these short divergence times, and we even have recent admixture. And so you get these patterns where you have lots of, the only private variants are rare, and the only most private variants are rare. And then once something is common, it tends to be found in a shared configuration versus a private configuration. Okay, so that kind of undermines the idea these continental regions have much information about them, because like when something is private to a continent, it's usually super rare within that continent. So what's the continental ancestry category doing for you, really, right? Great. So here's a less granular view. So looking at 26 populations, this is 100 variants grabbed at random and plotting the variant frequencies in a kind of color scale. And you see this general pattern of lots of rare private variants, common variants that are shared, and a kind of a little, kind of background in between where things have started to get a little more common and spread between populations connected by gene flow. And then this is the same view with that sort of discretization technique. Again, rare variants that are localized, and then when something's common, it tends to be found across the world. Super neat. Okay, so that's this pair of ideas. GGB is that Geographic Genetic Variance Browser. We call these GeoVar plots, that are this tool for looking at this broad geographic patterns of the variant set. You can think of it as a visualization of a 5-way frequency spectra. If you're into like thinking in those terms, it's like, that's a hard thing to visualize, but basically what we're doing is discretizing it and then showing the relative mass in each of these like quadrants of the frequency spectrum in a stacked bar chart. We hope that these provide a new view of population structure that conveys these typical allele frequencies. And it's a reminder that many of the things we use day to day, they don't convey absolute patterns of differentiation. And I know like many folks in here, might work on more conservation genomics applications. A classic one in conservation genomics, like, yes, I see structure, but is that structure sort of meaningful in a sense, right? How deep is the divergences? That should play some role in thinking about the management and not just the like existence of the structure itself, right? We hope these can be used to address misconceptions that arise often from like genetic ancestry testing, right? So I've already made that first point, but like, in genetic ancestry testing where, 23andMe, these various companies are telling you about what proportion of your ancestry is from different parts of the world. You might think for that to work, there must be lots of variants that are like specific markers of one type of ancestry, but that's just not what, human variation shows. These methods work by statistically aggregating lots of weak signals, subtle allele frequency variation, right? And then there's a point that I didn't make with these plots, but it's in the paper about when individuals differ, it's due to from the other. It's most often due to common variants, because like for Andy and I to differ, it's like, it's most likely that one of us carries a common variant and the other one doesn't. It's unlikely that one of us carries a rare variant because they're so rare, right? So even the ways that like Andy and I differ are likely a common variant that could be the way that any two people across the world differ because those common variants are so widespread. All right, and then ultimately, hopefully this helps calibrate expectations in terms of, for people doing GWAS studies in humans, for instance, that if you find a GWAS variant that's common, it's most likely going to be found in other populations. And if it's very rare, maybe less so, right? So, okay, so that's sort of this first chunk of a problem we've been working on. Not terribly sophisticated in terms of modeling at all. It's just about like, let's think about some drawbacks of current approaches and try to develop some interesting visualization approaches that address them. So this next problem is about whether one should sample broadly or narrowly and like what the consequences are of that in human genetics studies. So right now in human genetics, there's a big movement towards using biobanks. So these are large scale projects that are bigger than genetics and genomics in terms of like looking at health outcomes for large groups of people. So one of the chief examples is the UK Biobank, and this is a sort of a representation of from their work of like all the different phenotypes and medical records and so on that are being collected on these individuals, and they're available for researchers to try to make associations between genotype and phenotype. But there's a good question, if we have these biobanks that go very deeply in a few places like the UK, there's a Japanese biobank, there's the Finnish one, Chinese biobank that's being formed. The US is now building the All of Us biobank. And what are the consequences of going in a few places very deeply versus sort of spreading that sampling effort out more widely, right? And there's a lot of discussion of sort of, well, how can genomics be more equitable for global populations? And so there's a question of like how to understand this in more than just like sort of verbal positing, and like what can mathematical models bring to bear in terms of thinking about this. So in thinking about this, we need to think about a few things. One of them is that alleles with large effects, the ones that might be the most like instructive to us about pathway biology, they tend to be rare. So this is a plot of minor allele frequency on the x-axis for variants found to be associated with human stature, human height. And this is the effect sizes on the y-axis. And there's a notable, the red line, like below this, in this area are sort of things that are so small or rare that they're undetectable. And so the noticeable thing about this plot is the absence out here, that there are not many high minor allele frequency variants that have large effects, okay? The large effect variants are found only when you get to very low frequencies. That's a signature of some kind of stabilizing selection, that the large effect variants are very rare, some kind of negative selection acting on those alleles. And so that's kind of observation one. Alleles with large effects are things that might be really instructive, like big perturbations to the pathways and the biologies are rare. Then already from the previous part, we have this point that rare variants should be localized, right? They're young, they tend to be geographically clustered near where they arose. So this is a cartoon of like different populations connected by migration. An allele arises here, it spreads locally within that deme, then to neighboring demes. And only after a long time does it become more widespread. And so that's what we think a lot of these things are, is like just recent local mutations. So keeping that in mind, what are the consequences of this, of sampling broadly or narrowly? And here's a cartoon of thinking about this problem. So in this panel here, I've got a bunch of rare variants that are like, so this is a single variant that's got several carriers today. And here's another one, and so on and so forth. And so we're saying, like, thinking of these as deleterious variants that have this clustered geographic distribution. And the goal is maybe to try to find these, because each of them is an interesting perturbation that human biology might teach us about how we work and how to design a therapeutic for a particular disease condition. And if we do broad sampling, thought of here is like sort of thinking about like a sampling according to a Gaussian distribution that has a center, and its scale is either broad or narrow. how many do you end up detecting? So in this cartoon here, you would detect, variants maybe across a wide range. And here with this narrow sample, you detect variants across a very, short area. But the allele counts, what's that? Oh my gosh, thanks for clarifying that. Oh my gosh, okay. Okay, good. Good. You interpolate and impute. Okay, so great. Okay, yeah. So, right, broad area, narrow area. And notice like the number of variants and the allele counts in which they're found in, it might differ between these different strategies, right? And so how can we come to understand that? And so this is a problem that requires modeling the spatial spread of these not neutral variants, but deleterious variants, okay? And it's the moment for me to appreciate that a lot of modeling has been done of the spread of advantageous variants, going back quite deeply. This is a figure from a review that we're writing. It's kind of review on the spatial spread of adaptive alleles. And there are early models of what was called the wave of advance of how a mutation arises locally and then spreads outwards here in a one-dimensional habitat, published as early as 1937. And then, here at University of Oregon, there's been some of the best work that's been done in this problem area. It's been done by Peter Ralph and Jason Palus, and where you have multiple copies or independent origins of a favored allele spreading and eventually like tessellating, tiling the surface, or potentially having one take over all the different mutations. And then Jason's worked on models with long-range migration and documented this phenomenon, this core versus halo pattern that happens because the long-distance jumps establish these sort of satellites that then spread and converge into the main or create new centers. So super fascinating dynamics and really important. And we actually, so this review is sort of thinking about these models in the light of like the spread of Omicron, or like maybe population genetics should be thinking more about these kinds of problems. And there's a lot more work to be done in those areas. But going back to deleterious alleles and how they spread. What I want to show is results from working on this project, and Dan Rice in particular has been the kind of spearhead on this project, and he's been working with Christian Porras, a former undergraduate student, and now Maggie Steiner in my group is sort of taking over this project and pushing it forward. And so complicated problem because you have spatial structure, right? You have drift. You have selection, you have mutation, because you want to think about like these mutations coming out into the population and then dropping out. And then we're going to have variable sampling intensity. So quite a number of different pieces in play here. And Dan's been able to work out a form for the site frequency spectrum in these models. And so here what I'm showing on the right is the expected site frequency spectra, so the number of mutations, this is site frequency spectra of the deleterious alleles. And so like neutral mutations would show a -1 slope here, but you have this kind of, all these have this concavity where there's fewer common variants relative to the rare variants because of the selection that's acting. But the 4 different lines here, you can see the shape is changing as a function of the spatial dispersion of the sample. So the black line is if we sample just one location in the habitat and put all our sampling effort there. The 100 is sort of at the other extreme where we stretch our effort out, okay? And we see that if we have a very broad sampling configuration, we will have many of the variants that we discover will be at low minor allele frequency counts. And if we make point sampling, we get variants that are oftentimes with much higher copy numbers. Okay. The shape here is interesting in that it has this form that is much like the form for negative selection that you would get in a single population. But you have these scale parameters, such as this A parameter and the ZC, that depend on the migration rates and the sampling scale, so that are sort of shaping how quickly this exponential killing term works to kind of cut this thing off, and then also the sort of scaling at the boundary. So we're getting some insight into these areas. So there's a kind of discovery versus dilution. So words we're using here, that if you do broad sampling, you get more alleles in terms of like you're discovering many variants, but they're at very few copies. They're sort of diluted. Relative to narrow sampling, you might have a better chance of getting multiple copies. So depending on your application, you might favor one approach versus the other. And just to maybe in terms of background, finding multiple carriers of the same deleterious variant might be useful, because these days there's ideas of like what they call PheWAS, of let's look at the medical records of these people who carry this damaging variant and see what phenotypes they carry. And in that way, we'll learn what the role of a perturbation of that gene is on the system's biology, right? So, right. So if that's your goal and you're trying to detect these rare variants, then you'll find that if the broader the sample is, the higher the sampling effort would be. So this is relative to point sampling. As you increase that broadness of the sample, you increase the effort that you make to discover one of these rare damaging variants, and like add another carrier to your sample. Okay, but then there's a trade-off that, like the narrow sampling allows you to get multiple carriers per variant, but if you're trying to do prediction of like use these variants to predict like someone's heart risk or whatnot, like putting these into a polygenic score type model, there's a very interesting dynamics occur. So this is, let me introduce the plot. On the x-axis is distance based on where you sampled and going further and further away from that source, okay? And what's on the x-axis is the heritability explained, genetic variants explained in the focal deme relative to if you sampled it uniform. So if you sampled uniformly, then there's, no matter what distance you go on at, okay, like you will have the same level of explanatory power, okay? But as you start to, and that's sort of this baseline at 1. But as you start to make your sample more and more narrow, so going towards these purple colors, you start to do much better at explaining local populations versus distant ones, okay? And then what's neat too is this trade-off, like where this crossover occurs, can be understood in terms of like a particular characteristic scale that's given by the ratio of migration to selection. So if migration is large relative to selection, you'll need to go further out, like your explanatory power goes over a larger scale than if migration is weak, or conversely, if selection is weak, you get a larger scale at which your explanation's working. So, all this is to say, like the trade-offs in practice are like a complicated function of the migration rates and selection coefficients of the, the trait you're working in. But you might imagine, like for a trait that experiences like weaker selection, you're going to get a broader like transferability of your tools that you build for prediction, versus if you have a trait that's under more stabilizing selection. It's all these like local clustered variants, right? So yeah.
So I'm sorry to interrupt, but have you looked at neutral sites that are linked to these selected sites?
No. Yeah. So here we're not. Yeah. We could maybe do it in simulation, but we haven't gone to thinking about sort of linkage effects or that yet. Yeah.
Particularly from I love this perspective of thinking about prediction, portability, or think about portability of PTS or whatever. But fundamentally, we're looking at the neutral sites and not those that are under selection for those kinds of experiments generally.
in the sense of like we're looking at tag variants rather than the, yeah, I see. Yeah, yeah. This is thinking about like if we were building it off of the actual QTL causal. Yeah. So there's some ways in which it gets ameliorated by being at those tag sites. I'm unsure. Yeah. Yeah. It's a great question. Yeah. Great. So, oh, I see something in the chat. Okay, great. All right. So, okay. So this is very, yeah, preliminary work. The hint is that narrow sampling might be good for discovering pathways. We're finding these, you know, like getting many carriers at the same, you know, deleterious, like a loss of function variant that might, you know, you're not going to find many other copies of it, or, you know, and then you can, you know, characterize what that, you know, carriers of that mutation are, you know, experiencing. Broad sampling to get at this more equitable, like having better scales at which you can do prediction. But this is still early days, so we want to compare this to simulations. We want to assess robustness. We want to characterize these trade-offs in more precise terms in terms of like the like standard errors on effect size estimates and a relationship of genotype to phenotype, for instance. Examining saturation effects. So like this point sampling, you know, is like a lot of populations that you can do point sampling in, there's a, you know, there's a finite end that, you know, you might saturate in a way that these models don't capture well. We're still trying to get a grasp on that. And in particular, there's a lot of like deep efforts in founder populations, like Sardinia, like the Finnish, like, so there's, you know, how does that affect things? Want to look at some large scale data sets and empirically validate just sort of some of these predictions by playing, you know, in a well-sampled data set, we could play with subsampling the data to look at, you know, mimic focus sampling versus dispersed sampling. And then the methodology should extend to multi-point sampling. Like we have two foci of you know, two biobanks, you know, like what spatial scale do you actually, like the trough of prediction actually kind of not dip that much that you're sort of doing well enough. So, you know, highly abstract models, but I think they're helping like coach our thinking, which is one of the great values of these, of mathematical modeling in general, right? Okay, so moving on nicely. So now for this sort of third problem that we've been working on, I kind of bridge this two like topics of fine scale structure and then getting ancestry right for science and society. And this is a briefer piece. Oops. Okay. So, you know, what are fine scale patterns of human population genetic structure? This is a topic that my group has been involved in sort of addressing for a long time. It stems out of working my PhD in like interest in human variants and their, you know, one of my PhD projects was on the geographic distribution of this CCR5 delta 32 variant, and a lot of hypotheses that were quite colorful about, oh, maybe it was gave resistance to the plague or smallpox, or it was spread by Vikings. It's like, well, you know, if we want to understand that, we need to understand sort of the background history and structure and to be able to interpret those variants. And that kind of led in ways to working on spatial structure in European populations in this collaboration with Carlos Bustamante, Matt Nelson, and then into almost at the same time an interest in learning patterns of gene flow across landscapes, and this project with Matthew and Desi Petkova, and then this project where we applied these vector migration service approaches to large-scale samples for humans, and looked at how gene flow kind of looks across a landscape, so to speak. So this is the kind of a pair of pictures from the first and the third of those studies. So the first is this PCA map of Europe type result, where it's a PCA of Europe, and each two-letter code is a single individual, and then the median position of individuals with the same two-letter code is shown with the big dots. And the broad thing that you see is an alignment of the genetic variation in the sample with the geography of Europe. to be clear, that the samples here are labeled by grandparental ancestry and individuals who have like no conflicts in their grandparental ancestry. These individuals were like, as far as we know, all their grandparents are from Italy or Spain or Portugal. And so this is really like a reflection of structure in the genetic ancestry more than like on the ground. Most of these individuals were sampled in Lausanne, Switzerland or London, okay? But have these ancestries from across Europe, and that ancestry that has this structure, okay? But the key, what made this study exciting at the time and interesting was seeing such fine scale structure that is not that there's sort of some homogeneous group and that's the end of the story. There's fine scale structure and a scale, and that there's a sort of genes mirroring geography, sort of the punchline of that paper. And then here's the view from using this migration surface approach applied to samples from across Africa and Eurasia. And these areas of blue are areas of high effective migration. Areas that are darker are areas of low effective migration. These shouldn't be interpreted as like hard barriers. They're just areas where the gene flow is lower. Like we know that alleles move across these barriers, these like what's called troughs in migration. And so the punchline here in terms of this part of the talk is to emphasize how the continental structure that people bring to thinking about human variation is not very useful, right? That you have fine scale structure within continental regions. At the larger scale, you see all of these more wrinkles that are in places that align with a lot of like physical geographic features, but aren't exactly these boundaries of continents, right? There's a lot of it's neither like smooth geographic distance that's homogeneous across the whole globe, nor is it that there's chunked continents. It's something more complex than that and more regional. And so, with that in mind, sort of always been this tension of the way that people describe ancestry in human populations. And I was lucky, along with one of my postdocs, to be involved in this group. So Mashal Sohail in particular, to be involved in this group that was reflecting on how to describe genetic ancestry, and published a kind of perspective piece just recently. And some of the punchlines of that are, of course, and I think this is probably well appreciated by many people here, but like the value of using continuous ancestry variables where possible. Be careful about the slippage between continental ancestry and race. So that's one of the particularly like difficult things about using these continental categories, is people basically map like European ancestry and African ancestry to like white and black, and so trying to avoid that way of characterizing human variation. The kind of difficulty like in building diverse samples to kind of achieve this goal of equity, being careful not to reify race or imply that there are huge differences amongst human populations, because it is, more complex than that. From, hopefully you got that impression, especially from the first part of this talk, that sampling broadly will help you capture more variance and understand biology of more people across the globe. But it's not that there are these large numbers of alleles that are chunked into some parts of the world and not others, right? And even if you're not working in human genetics specifically, the important role that most of you are going to play as teachers in the classroom and teaching pre-meds and teaching about human genetic variation at some point, even if it's discussing your uncle's 23andMe results or something. But how to convey these topics well in the classroom. And so we've been trying at the University of Chicago to kind of inject more and more of it into our curriculum. And some of the things, to give some practical suggestions, are in those responsible conduct of research courses. We've injected we have like two versions of it, one more basic, one more advanced. The advanced one, we have two genetics and society sessions, one on eugenics and genetic engineering, and then the other on genetics, race, and discrimination. And happy to share like what we use for that. And then been trying to invite genetics and society speakers in our genetics seminar series. in our pop gen classes, we put in some classes reflecting on genetics and race and like the public discussion of it versus just sort of talking about how to measure population structure without thinking about the societal issues. So, so that, yeah, hopefully all of us can be better at sort of teaching these things and helping correct a lot of the very easy misconceptions that you can imagine arising given the proliferation of the way genetics is sort of becoming more common in society and medicine. Okay, so those are the topics I wanted to cover. I want to acknowledge the study participants and researchers generating and sharing the data that makes all this possible. I tried to thank people along the way, so I want to just kind of double thank them, the people I was mentioning along the way. And then a lot of the ambiance, the milieu at UChicago in terms of the different groups and faculty whose labs they get to bounce ideas off of. And yeah, reach out if you might be interested in joining us or collaborating. And that's it. Thanks.
Questions. And so I think you can raise your hand and just say something. Michelle. Is your meeting early right now? Good job.
Yeah, Zoom, can you guys hear us? Chat or thumbs up? Yes, okay, great.
Well,
Thanks.
they can hear you. You should ask if they can.
Oh,
Michelle.
can you hear Michelle? Yeah, I'll try to repeat the question. Okay.
First one, in your first section, maybe I hope this is like totally on track, but I was curious if in the distribution, some of those, I think by definition, it was pretty neutral, but I'm not sure. Can you unravel any linkage between some of those sequences that are common or not?
Okay, and relationship to sort of
Each
whether
other,
they're neutral.
in other words, are there linked sequences that tend to have the same distribution across geographic areas? This is the first section.
Yes, yeah, okay, great. Okay, so for Zoom, the question was about asking if there are just sets of variants or like linked variants that show similar patterns of, yeah, okay, great. And so different sets of variants are kind of enriched for some patterns versus others. We played with that, and there's some like, so yeah, you can take a list of variants on whatever criteria you want and ask like, well, what's the relative abundance of different geographic distributions of this set, and compare it to this genomic background, and that's kind of a fun thing to do. So like a nice one to do is the SNPs on genotyping arrays. Like what are their geographic patterns, and then contrast different genotyping array designs. So some arrays do much better at capturing variants that are found outside of Europe than others. Some have the kind of bias of that, like a lot of the early studies were, oversampling European variation, let's say. But I think you meant like more like a biological sense. And
yeah, there's allegedly kind of random sampling.
Yeah.
We're testing that.
Yeah. So this is it is just sort of, that was all the variants in the 1,000 genomes, like that random. A part that I skipped that I for time, but that's interesting, right, is like, what are, like the skin pigmentation variants, right? Or, some of these things that we see vary, yes, those are the variants that show like common in one part of the world or common, in certain regions and absent in others. And those are these ones where local adaptation has driven allele frequency variation. And of course, that's this, solves this conundrum of like, why do we have such superficial differences, but then, pictures like that one with the primates where you see how similar we are. It's that, right, there's been rapid evolution of like these superficial phenotypes, right? I mean, beyond, so yeah, in terms of linked, like looking at like pairwise, like LD in the geographic patterning, we haven't really looked at that. If that's,
There was a little bit of a
yeah. Yeah, you kind of, I mean, there should be some kind of correlation structure to these geographic patterns. Yeah.
The other question I had is much more explicit. In your, it's the same from the same section. You use the visual from National Geographic that really emphasizes the migration from northwestern Europe into Labrador, that area, the Canadian area, and yet, at least that's the entry to North America that was emphasized in that particular picture. But the kelp theory is now much more popular, 20,000 years old, would have been 20,000 year entry, and that should link Japan and even potentially some place like the Philippines to North America instead of Asia, instead of Europe or Africa.
Oh,
And
interesting.
also the very land bridge is a more popular.
Yeah.
And it's like 14,000 years ago, so it's more recent, and that would be Russian. So I was curious of both of that selection, but more, what it seems like your work should really help differentiate between these theories. Although they're probably all true.
Yeah. Okay. Thank you. So the question was about the National Geographic figure that I showed that appeared to show like connections between Europe and North America. Yeah. And given that there are many other alternative hypotheses, some that have a lot of support, and that was inadvertent. Like I'm not sure actually. That wasn't a willing choice there. I thought the figure showed it's intending to show the conventional, like what most human geneticists believe, which is most of the gene flow is over the Bering Land Strait, and I mean the land bridge, so yeah, so that was some accident of the way it displayed, or yeah, it's not that there's some new alternative like consensus. Yeah. But then the general point that like this should help us see migration is generally true. The goal here was more to just like show the structure in kind of a raw way and not fit any models, but you can start to see how, the fact that like those shared, at a very coarse level, shared variance between West Africa and African Americans living in the US, that's a signature of the movement across the Atlantic, right? And that on another level is a kind of way of seeing some of the migrations and demographic history. Yeah. I mean, I will say that a lot of the more, a lot of kind of detailed histories of gene flow and movement that are being revealed in many studies are often looking at like very fine scale differences in correlation between these common variants. And so that the coarseness of that discretization, you probably lose a lot of signal too for anything but these like big movements and big history. Yeah.
Great.
I was wondering if you could talk a little bit about how you think your work might be like conservation genomics. You mentioned that a little bit. You said candid evolution, but yeah, I was wondering if you could speak about that.
Yeah, thank you. So the question was about like applications to conservation genomics, and right. So there, one of the big themes in my course were like understanding this, genetic structure on the landscape, which is sort of like a starting point for doing conservation work. And we've been trying to develop out those migration, surface approaches and yeah, I apologize. I didn't speak about that today because I know that sort of many of the groups here have been like sort of reading those papers and know them quite well. So I want to talk on some of these different areas. But that's, I think, one of the big areas where I see my group kind of contributing in that space. But then I also like the sort of feedback of working in humans like that kind of pushed me to think about these variant-centric approaches. I hope that that's sort of influential for people in thinking about like looking at their, whatever their species of interest is in a more like raw kind of, because I think there can be a bit of a pipeline of people like, okay, I'm going to push my stuff through these classic pieces of software, get these classic set of figures that everybody has in their talks, but not actually like understand the genetic data in the gritty, kind of more visceral, like what is the actual allele frequency variation like on a landscape way. So, so I hope it was, useful in that way. Yeah. And then there are like spin-off things like the locator type projects. I have wondered about like, those flexible CNNs may accomplish what one could think about doing with these migration models, which is, assigning individuals on the maps. And the vanilla way to do that sort of in a non-locator world would be to like use some kind of Gaussian process that assumes like a generally they use like what's called spatially homogeneous covariance kernel, and this would give you a suggestion of how to make a heterogeneous kernel. Like there are places where the isolation by distance drops off quicker than others, and you might want to think about incorporating that if you're doing these assignment problems. But we haven't ourselves gone there yet.
Given the time, we should probably thank Jonathan.
Thank you.
And we're not having a tea time right after this.
Thanks everyone on Zoom.